Hello, I am Natasha Brown, and I am the program manager for the JGI-UC Merced Summer Internship Program here at the Joint Genome Institute. We're currently in our 9th year of hosting interns, both graduate and undergraduate students from UC Merced. And today I have the pleasure of introducing 3 of our outstanding interns today. Today we will be hearing from Tyrome Sweet, who is a 4th-year graduate student in the Quantitative and Systems Biology program, followed by Natalie Meachum, who is a 2nd-year graduate student in the Applied Mathematics program. And last but not least, we will hear from Edward Liu, who is in his 3rd year of undergraduate studies, majoring in applied math with an emphasis in computational and data sciences. So without further ado, I would love to pass the torch to Tyrome. Please take it away.
Awesome. So good morning. Today I'm here to talk to you about plant-associated and non-plant-associated bacteriophages. Next slide, please. Historically, there's been a focus on bacterial and fungal components in the rhizosphere, but not much is known about the bacteriophages in these systems. Bacteriophages are viruses that infect and replicate in bacteria and are an important component of the root microbiome. Next slide, please. Current literature has shown that lytic phages can impact bacterial abundance and composition during colonization of host plant leaves. Next slide, please. In 2017, Ben Cole and others conducted an RBT and seq analysis, and they found a set of 115 genes that were deemed to be essential for root colonization by Pseudomonas simiae. Of these 115 genes, there was a category called Other, which included phage genes, later verified by Janelle Basso. Next slide, please. This shows that there may be a connection between the plant, bacteria, and phages. The core of this work is to identify whether phages were truly modulating the ability of Pseudomonas simiae to colonize plant roots. Next slide, please. So our research objectives were to detect phages and select plant-associated and non-plant-associated bacteria, determine the impact of these phages discovered, and to identify phage homologs. Here's how I did it. Next slide, please. I used the pipeline of my multiple plant-associated and non-plant-associated whole genome sequences. I used CheckV to help minimize false positives. I identified potential genes using DROM-V to get a better understanding of the identified phage's impact, and I used phylogenetic tools to determine any similarities or differences in the plant-associated and non-plant-associated phage. Next slide, please. Maureen Berg performed the initial Beer Sorter 2 analysis on 3 bacterial species, where she found a high confidence of identified phage in Pseudomonas simiae and Pseudomonas petita. For Pseudomonas simiae, we see the location of the 3 phage signatures along the genome. Now, using the full data set of 10 genomes, I observed, next slide, please, that double-stranded DNA phages were observed in all host genomes. And as an example, Pseudomonas simiae had 3 phage regions detected, which confirmed the preliminary and experimental results. For the non-plant-associated bacterial genomes, we've seen that, next slide, please. We saw 1 bacterial sample with no phage detected. And like the plant-associated phages identified, these were also all double-stranded DNA phages. Next slide, please. The Beer Sorter 2 data, not shown here, gave high confidence levels that range from 0.82 to 0.96. So CheckV was used as a form of quality assessment. Here, phage quality is on the x-axis, while the number of phage is on the y-axis. We observed 3 complete and 25 partial phage signatures respectively, of which 5 were high quality phages. Next slide, please. Idle and cluster omega were used to show phylogenetic association of the identified viral signatures. Green denotes plant-associated phages, while red denotes non-plant-associated phages. The viral signatures are labeled according to their respective bacterial host. Next slide, please. Our sample size was small, so we can't definitively say there was a clear association, but given the current results, we can use a larger sample size and annotate the viral signatures to make a more definitive conclusion. Next slide, please. So in the future, I hope to use the pipeline to annotate the plant-associated and non-plant-associated phage using JGI-V to determine the impact of phages in each of their hosts. So with that, next slide, please. I just want to thank everyone on this slide and all those who helped me with this project. And most of all, thank you for listening to my presentation. Are there any questions?
So actually, for these flash talks, we can't take any questions since I'm doing the slides as well. So I'm afraid we just have to move on to the next talk, which is Natalie's talk. And we can get any questions to Tyrone if you have them. Sorry about that.
Awesome. Thank you.
Yeah. Okay. Natalie, could you sign on, please?
I'm here.
All right, go for it.
All right. Hi, everyone. My name is Natalie. I'm a third-year applied math PhD student at UC Merced. This summer, my mentor and I worked on applying pangenome graphs to genetic variation in plants. Next slide. Thanks. So the title should be there. I don't know why it's not, sorry. But the question is, what is a pangenome? A pangenome is a collection of genetic information from many genomes, 2 or more genomes. So a pangenome will consist of core and variable genomes, meaning there are parts where the genomes overlap completely, parts where they don't overlap at all, and parts where some genomes overlap and some don't. Next slide. Why pangenome graphs matter as an approach is that previously a lot of science was done by mapping subject genomes onto reference genomes. The problem with that is something called reference bias. So in the image, we see that if our subject genome has an insertion or a new chunk of information that maybe lengthens the genome, on the reference genome, there is no place to put that insertion. And so when you look at the mapping, it's kind of as if that insertion just doesn't exist and we lose that information. So by using pangenome graphs, we are able to avoid reference bias. Next slide. Here's an example from 3 chloroplast genomes, which was kind of the first part of my summer. So we have 3 different genomes for 3 different trees. And the main point of this is to show you the image and kind of explain what it represents. So if you look at the beige part, that is where all 3 sequences are the same or essentially the same. Meanwhile, we can see that blue, the walnut tree, is more different from the rest because it splinters off early. So we have this big bubble where walnut is different. And then in the yellow, we see that the other 2 genomes are much more similar. They go along on the same path for a while, but then they do splinter off into those green and red bubbles. So I used minigraph to construct the text version of these graphs and then bandage to visualize them. There is a caveat here, which is that the bubbles are not necessarily as different as minigraph says they are, but that is just something to keep in mind for future work. Next slide. So the central project this summer was to look at somatic variations in a modern day lemon. So here's the family tree for that lemon. We can see that 2 of its grandparents are pomelo, which is similar to a grapefruit, and then mandarin. Those 2 give rise to sour orange, and then sour orange and citron each contribute one side of the genome for the lemon. So in this case, we're interested in everything that's green. So we're looking at the combination dark green, light green genome aka what comes from the Pomelo and Mandarin ancestry in the lemon. Next slide. So when I talk about somatic variations, I'm talking about the white lines in this picture. So when we compare the sour orange Mandarin genome up top to the sour orange Pomelo genome on the bottom, and then in turn, what that lemon genome looks like today. The somatic variations are the white lines. They come from mutations during clonal propagations. So it's kind of, for example, what has changed in the lemon since it was propagated as compared to what has changed in those same genes from, for example, the pomelo. So we're comparing dark green to dark green, light green to light green. And if we compare light green to dark green, then we're comparing two different species. Somatic variations are important because they matter a lot when generating phenotypic diversity. So if you're at the grocery store shopping for oranges and you have a lot of variety available to you, like especially if you're at Berkeley Bull or something, that variety is largely available thanks to somatic variations. So my task this summer was to study those in lemon, in particular comparing lemon and sour orange, and find a way to characterize those variations. Next slide. Thank you. So to characterize the variations, we first said, if we're comparing two sequences, how similar in length are they? And then if they are a similar length, are the sequences being compared very similar or very different? And so by kind of sorting those into these categories, we were able to find that the vast majority of somatic variations are some form of deletions or some form of insertions. Next slide. Similarly, I mentioned we could also compare dark green to light green. So those are interspecific variations between Mandarin and Pomelo. And if you look at the non-somatic and somatic columns, you can see that as expected, because we're comparing two different species, we have a lot more non-somatic variations than somatic variations. Next slide. Finally, we were able to take a deeper dive and look at repetitive sequence types in particular. So long terminal repeats are really important here. And we were actually able to conclude, if you look at, and note that this graph is actually log scaled. So if you look at all of the labels at the bottom on the left, we can see that the vast majority of repetitive sequence types are actually long terminal repeat retrotransposons. So we can conclude that those are the dominant source of somatic variation. Next slide, please. So at the end of the summer, here's what I knew. Pangenome graphs are an amazing tool to characterize genetic variations efficiently and without reference bias. Minigraph is able to identify large bubbles with similar sequence length. There is an issue to analyze further about what exactly similar may mean here. And then finally, LTR retrotransposons, we can say, are likely the dominant source of natural somatic variation in plants, as shown through the lemon ancestry. Next slide. All right. Thank you to everybody who contributed to this summer. Thanks.
Thank you very much, Natalie. That was great. All right, a final of our flash talks is from Edward. Edward, can you unmute yourself and join us, please?
All right. Hi, everyone. My name is Edward, and this summer my project was studying Hemimastigophora, and we were fortunate enough to find a potential new lineage. Next slide, please. So this is our methods, and so basically what we're looking for were bidirectional best hits with the Hemimastigophora. So our procedure was we would start with our metagenome samples, and then we would blast it versus the known Hemimastigophora transcriptomes. After extracting those hits, we would then reblast it against the NCBI-NR database. And if it returned with Hemimastigophora again, then we've confirmed that we got a bidirectional best hit, which has a very which is very certain that it's going to be a hemimastidophora sequence. Next, we tracked back those contigs that are bidirectional best hits, used BUSCO and UCC to check the completion of those samples. And then finally, we chose 3 of the most common proteins to build a phylogenetic tree using MAFT, AMAS, and IQ-tree. Next slide, please. So this is a graph that shows many things. First of all, you can see on the bottom the sizes of our bins. So we have many bins. Most of our bins are small, but we have many bins that are over 1 million base pairs, and that was really helpful for our analysis. In addition, you can also see the number of proteins corresponding to the base pairs, as well as the completeness. Next slide, please. So these are our results. So we used an outgroup of Volvox, which is a green algae, Physcomitrium, which is a green moss, and Arabidopsis, which is a flowering plant. And we use that to build our phylogenetic tree. So what was most exciting is that you can see that there is a new separate branch that differs from the known Spironema and Hemimastix transcriptomes. And that separation has a bootstrap score of 81, which means it's significant. And yeah, that's the most exciting thing. And also you can notice that I've labeled all of the samples we have based on where they were collected from. And most of our samples were from soil, but our marine sample was very different from all of our soil samples. And that was very interesting as well. Next slide, please. Wait, is that the last slide? All right, yeah, so thank you for JGI because JGI made all of this possible. And also thank you to Miguel and Frederick.